Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 10th December 2020. And as all of us know, we are revising an all important chapter number 14, respiration in plants, and as part of it we appreciate it, we reflect upon, and we value this because in every cell that is present in our body all these things are happening, and we are becoming energetic, we are getting energy because of this. And it is apt that we should know how we are getting energy, how every cell, every tissue, every organ, and as an organism in total, how we get energy. And as all of us know, it is because of the food that we eat. So there is a saying, we eat to live, and we live for what? To eat. We live to eat. And as being biology boys, we know that we are eating something nutritive so that we get energy, and because of that energy we live, and we live happily. So always we eat to live, don't forget, but don't say we live for what? To eat. Okay, we are not for living to eat, rather we eat so that we live healthily. And how is that? When the food that we are eating is selective, like I have been telling you, your food should be always rich in proteins, moderate in carbohydrates, and few in fats, but don't avoid it. But along with that, you need to have vitamins and minerals, and most important, 8 glasses of water, 4 to 5 liters of water you must drink daily. Then you will be healthy, and there is meaning in learning about this chapter. Okay, and we know that whatever we eat, breakfast, lunch, dinner, snacks, all that, finally the end product of all this is glucose. And the first step is glycolysis. Gluco is glucose, lysis means breaking. Glucose molecule is broken down. Where is it happening? Inside the cell, in the cytoplasm. So when that happens, how many molecules are we getting? 8 ATP molecules of energy we are getting. And then it moves to mitochondria. In mitochondrial complex, the next part of the catabolism takes place, that is glucose molecule is being broken down into pyruvate, and pyruvate enters into the great Krebs cycle or TCA cycle or citric acid cycle. All three are named same, and then undergoes a lot of changes, as we discussed yesterday. Series of changes it undergoes and gives us how many molecules of ATP? If I am correct, 24 ATP molecules. So one molecule of glucose is giving us 8 ATP in glycolysis, 24 in Krebs cycle, 24 plus 8, 32 molecules, ATP molecules. So since both of them are giving energy, we appreciate it. So how is that we are getting the 24 ATPs, and what is the overall reaction of Krebs cycle? I am sure every one of you must have practiced at least once this Krebs cycle, because you need to remember all the chemical names, right from pyruvic acid, acetyl CoA, citrate, isocitrate, cis-aconitite, oxalosuccinate, ketoglutarate, succinyl CoA, succinic acid, fumaric acid, malic acid, and of course oxaloacetic acid. And oxaloacetic acid, having 4 carbons, combines with 2 carbon acetyl CoA and forms citric acid. That's why it is called citric acid cycle. So this cycle keeps on repeating as long as the acetyl CoA is there. Hence, this, how many times it gets repeated, that many 24 ATP molecules we get. Every mitochondrial complex will get in our cell. So basically, to revive tricarboxylic acid cycle or Krebs cycle. So TCA cycle was discovered by Hans Krebs in 1940. This cycle is called TCA cycle because initial product is citric acid. Acetyl CoA combined with oxaloacetic acid and water to yield citric acid in presence of an enzyme, citrate synthetase. How to remember the enzyme name? Here see, What is the end product? Citric acid. Citric acid, citrate synthesis. Citric acid is synthesizing, so citrate synthase to release CoA. Citrate is then isomerized to isocitrate. It is followed by 2 successive steps of decarboxylation, leading to the formation of alpha ketoglutaric acid and then succinyl CoA. In the remaining steps, succinyl CoA is oxidized to OAA, oxaloacetic acid, allowing the cycle to continue. There are 3 points in the cycle where NAD plus is reduced to NADH2 and 1 point where FAD is reduced to FADH2. These are very powerful energetic molecules. A molecule of glucose produces 2 molecules of NADH2, 2 ATP and 2 pyruvate while undergoing glycolysis. Okay, that we know it. The 2 molecules of pyruvate are completely degraded in Krebs cycle to form 2 molecules of ATP, 8 molecules of NADH2 and 2 molecules of FADH2. So overall the reaction will be I can say 2 acetyl CoA plus 6 H2O plus 6 NAD plus 2 FAD, 2 GDP, 2 inorganic phosphate gives rise to 4 CO2 plus 2 H2O. Look at that, glucose is getting oxidized to form carbon dioxide and water, and 6 NAD plus becoming 6 NADH and 6H plus, and 2 FAD is becoming 2 FADH2 and 2 GDP becoming 2 GTP, and of course 2 coenzymes, coenzyme A. And as I said yesterday, being catabolic, Krebs cycle provides number of intermediates which are used in different anabolic pathways to form important biomolecules like glutamic acid, aspartic acid. Hence this cycle is called amphibolic cycle or amphibolic in nature. Amphibolic pathway is the one which is used both for catabolism as well as anabolism reactions. Catabolism, breaking down; anabolism, making up. Okay, these reactions. And once this happens in mitochondrial complex, now what's happening next? Whatever electrons and protons that are formed in the process, what about their fate? So the fate of electrons and protons are to be decided, and that will take place in another step, step number 3. Step number 1, glycolysis. Step number 2, Krebs cycle. Step number 3, electron transport chain. We call it ETC, electron transport chain. And that is also terminal oxidation, last phase of oxidation. Is the name of oxidation found in aerobic respiration that occurs towards the end of catabolic process and involves the passage of both electrons and protons of reduced coenzyme to oxygen to produce water. Now, how does that happen? This is how the electron chain will take, ETC. So the inner mitochondrial membrane. See, there it is. Krebs cycle has taken place in mitochondrial matrix. Now the electrons and protons are moving to inner membrane of the mitochondria. It has groups of several protons, H plus, and electron E minus acceptors. These groups are arranged in specific series called ETC. You can see the diagram, the protons, H plus, and the electrons, where we are writing E. Look at that, E for electrons. Look at that. So it is arrangement there. That's why we call it as ETC, electron transport chain, or ETS, electron transport system. Now what's happening here? Now the complex name, like we put the table in the form. So NADH coenzyme Q reductase or NADH dehydrogenase complex. So you got inner membrane space, inner mitochondrial membrane, and matrix. So NADH is becoming NAD plus, and then FMN is taking place, and then protons H plus are released. So in this complex, NADH dehydrogenase and FMN and FES center. Okay, and 2 protons and 2 electrons from NADH plus H plus are transferred to FMN. You are seeing here 2 electrons and 2 protons are transferred to FMN. Immediately after getting these electrons and protons, the FMN gets reduced to FMNH2. Electrons are then transferred from FMNH2 to coenzyme Q, quinine, coenzyme Q. And this is also called UQ, that is ubiquinone. You are seeing here UQ, ubiquinone, and this gets reduced to CoQH2. You are seeing here CoQH2, and then during this process 2H plus are transferred from the matrix side of inner membrane of mitochondria to the intermembranal space, and then succinate coenzyme Q reductase, or it is called the succinate dehydrogenase complex. It includes the membrane bound enzyme of TCA cycle and succinate dehydrogenase FAD and FES center. 2 hydrogens removed from succinate reduce FAD to FADH2. From FADH2, the electrons are transferred to CoQ via FES center. This one, FES center, 2 electrons are transferred. And CoA is reduced to CoQH2, that is CoQH2. And then the third complex, cytochrome C reductase complex. It consists of cytochrome B, Reski FES center, and cytochrome C. As you are seeing here, you have got cytochrome B center, cytochrome C center. Okay, so this is the third complex. Electrons donated by CoQH2 are transferred through cytochrome B. As you are seeing, they are transferred to cytochrome B, FES center. You are seeing 2 electrons getting transferred, and cytochrome C1 to cytochrome C, cytochrome C1 to cytochrome C, which gets reduced, and 4 protons are transferred from matrix to intermembrane space, as you are seeing. And like CoQ, cytochrome C is a mobile electron carrier and is not part of any complex mentioned there. And then last complex, cytochrome C oxidase complex. So here it includes cytochrome A, cytochrome A3 and 2 copper ions, okay, Cu2 plus copper ions named A and B. Electrons are received from cytochrome C when gets oxidized, and electrons are ultimately transferred to oxygen to form water. So the purpose is formation of water. This step is called terminal oxidation. So if you see in the electron transport chain, totally there are 4 complexes. Complex number 1, NADH coenzyme Q reductase, or in short we can say NADH dehydrogenase complex. That is complex number 1. Second one is succinate complex, or succinate coenzyme Q reductase complex. Third one is cytochrome C reductase complex. Fourth one is cytochrome C oxidase complex. So in this chain, electron transport chain or electron transport system, there are totally 4 complexes, and at the end of fourth complex, the electrons that are formed will be ultimately transferred to oxygen, and the final product of water is formed. This is the purpose. So there is a 5th complex, that is ATP synthase complex, which is involved in the preparation of ATP synthesis. Okay, that we'll talk separately. Now, so far, what is that we have done? Electron transport chain, ETC or ETS, the metabolic pathway through which the electrons passes from one carrier to another inside the inner mitochondrial membrane is called ETC or mitochondrial respiratory chain. Electrons from NADH produced during citric acid cycle are oxidized by NADH dehydrogenase, and electrons are transferred to ubiquinone located within the inner membrane. Ubiquinone also receives electrons from FADH2, which is transferred to cytochrome C via cytochrome BC1 complex. When the electrons pass from one carrier to another via electron transport chain, they produce ATP from ADP and inorganic phosphate. The number of ATP molecules synthesized depends upon electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP. This is important. 1 molecule of NADH, while oxidation of 1 molecule of FAD produce 2 molecules of ATP. So this is the overall picture of electron transport chain. So since all the 3 steps give a lot of energy, it is important that all the 3 steps together comprehensively you need to study. Then you understand by oxidation of one glucose molecule how many ATPs are produced. So keep on studying, understand it as a together, the whole concept. All the 3 steps together you must understand. That is glycolysis,